Have you ever heard of the phrase, if you hear hoofbeats, think of horses, not zebras? Well, this sentence is commonly used in medicine to point out to students that the most obvious solution is usually the right one. But while zebras might not be very common in medicine, studying them, studying the unusual solution, can actually lead to some very unexpected findings. And let me start on a personal note. Back in 2013, when I had just finished my studies in biomolecular engineering, I was in a meeting with my professor when he suggested, "Hey, Doreen, I think you should do your thesis either on antibodies from cows or from sharks." And I was like, wait, what? Seriously? Did he just say cows and sharks? Because after all, that's not necessarily what came to my mind when I was thinking of cancer research and healthcare, to be honest. But he was right. Antibodies are the guardians of our immune system. And it turns out that some animals had to evolve unusual solutions in the form of molecules in order to maintain a functional immune response that would protect them from diseases in their environment. And these unusual molecules are special types of antibodies. And just as a one-sentence refresher, antibodies are a class of proteins that the immune system utilizes to fight bacteria, viruses, and other pathogens. And we have them, and like any other animal, sharks have them as well. And to come back to 2013, I ultimately ended up doing a PhD investigating shark antibodies. And that is what I want to share with you today. There are unusual molecules in nature, and we can engineer them for our purposes. Now, although many among us have probably never seen a real one, sharks are known to all of us for many attributes, but most likely not for their fancy immune system, right? However, these animals have evolved a molecular defense mechanism that has unique characteristics. First, sharks inhabit marine seawaters that have high salt concentrations. And they have adapted over time to avoid dehydration due to severe loss of water upon enriching urea in their blood. Urea, though, would normally destroy proteins such as antibodies, and that would be not so good for the shark. So, this species has evolved incredibly robust antibodies that are structurally very stable in order to maintain a functional immune response. Secondly, shark antibodies are actually so robust that when you heat them up to very high temperatures, say around 90 degrees Celsius, they will completely lose their structural integrity and become unfunctional. But if you subsequently cool them down, they will just jump back right into the original structure and do their job. And that's pretty neat, isn't it? And no, before you ask, are human antibodies? Unfortunately, cannot do this. And third, while human antibodies have a rather flat surface and therefore interact with rather flat proteins, shark antibodies have more rugged structures that are able to bind to very recessed and cleft-like spaces on their interaction proteins. And this means, basically, that shark antibodies can easily reach places where human antibodies are often at loss, such as virus particles or toxins, for instance. So those are all quite unique characteristics. Structural stability, capacity to jump back from functional loss, and the capacity to easily interact with complex molecules. But how does this help research? And in particular, our fight against cancer. One of the challenges when developing a new anti-cancer drug is that we need to be aware of the path that drug takes within our body. We need to thoroughly monitor it so we know what's happening in the body and we can prevent unwanted side effects. Now, many of the most promising anti-cancer drugs that are in the market are, in fact, so-called human therapeutic antibodies. And these are specific proteins that can be administered to patients to attack a tumor in their body. Most of these therapeutic antibodies are human, which means they're tolerated really well. In fact, many of them will blend right into our immune system and support it in the fight against the disease by binding to mutated cancer cells. However, when you want to track down the anti-cancer antibody, the fact that it's human is a problem. Think about it. You have a perfectly human anti-cancer antibody designed to support your immune system. But now you need to track it down in the human blood that itself contains millions of your own natural antibodies. And it's difficult because indeed your own antibodies and the anti-cancer drug look and behave almost identically. But this is when our shark molecules can come in handy. Because of their unique ability, as I've said, to bind very complex molecules, shark antibodies can actually grab the anti-cancer drug, and most importantly, specific parts of it, and help us isolate the anti-cancer drug from human blood serum. And since every human antibody, natural or therapeutic, has a different and unique region that interacts with a specific target, such as the cancer cell, a different fingerprint, if you will, shark antibodies can only recognize one particular fingerprint, the anti-cancer drug, and don't interact with any other antibodies, making them really specific. To summarize it in a very simplified way, it becomes easy to find the therapeutic drug in the blood because we have an engineered shark antibody that specifically captures it. Shark antibodies seem to be predestined for this job because, in contrast to existing methods, it's so easy and quick to generate them. Now, we don't actually engineer the whole molecule, but just a part of it that has those specific features. But the best part is, we don't even need a shark for all of that. Now, what we do is we mutate the shark antibodies in a reagent tube about this small, and we create a huge diversity of different variants. We call that a shark antibody library. We then add the human anti-cancer drug to this library, and we go through several selection rounds in order to identify the perfect match of a shark antibody and the anti-cancer drug. And once we have identified these matching molecules, it becomes very easy to track down the therapeutic drug in the blood. I should add, for clarity, that we do not plan on injecting shark antibodies into patients. We would simply use them when analyzing the patient's blood in the lab. But upon using shark antibodies, we can actually characterize newly discovered anti-cancer drugs, so we know everything we need to know before these drugs proceed into the clinic. So you see, a shark antibody can actually help in developing the next generation of anti-cancer drugs. In addition, the high thermal stability and simple yet robust structure of these molecules makes them ideal for many other medically relevant applications. We could envision to use them as detection reagents when testing for a disease, for instance. Shark antibodies can tolerate really harsh environments since they function in the shark blood. And since their structure is so simple, when compared to other reagents that are commonly used for such diagnostic tests, they're easy to manufacture. For instance, we have a problem with the storage of diagnostic tests in countries where surrounding temperatures regularly rise above 40 degrees Celsius. We already know that shark antibodies can address very complex proteins such as virus particles or toxins. So why not use them for diagnostic tests for neglected diseases? Using super-stable shark antibodies could increase the test's shelf life and improve medical care in many underserved areas. And as I briefly mentioned in the beginning, it's not only sharks that offer unexpected and unique antibodies. We can find analogous molecules in many other species, each of them having very special traits. We can explore them, we can harness them, and we can engineer them for our needs. Now, I don't know of any specific research into zebra antibodies yet. But I would like to encourage you, the next time that you hear hoofbeats, you might want to think of zebras, or maybe sharks, instead of horses. Thank you.